Thank you all very much. Thanks for the nice introduction, Tara. It's nice to see you all here on a warm, bright, sunny day, sitting inside, enjoying a cup of coffee. As Tara said, my name is Peter Girguis. I'm an assistant professor of microbiology here in the OEB department at Harvard. To give you a little bit more information about myself, I think of myself as kind of the auto mechanic of biology. And what I mean by that is I'm a physiologist at heart. I'm really interested in understanding how things work and in understanding how their capacity enables organisms, in this case microbes, to make a living in our environment. And here at Harvard, we've been doing a lot of work on putting a lot of effort into studying what I'm going to call uncultivated microbes. Now, who here has cultured microbes before? Well, all right. And it may surprise you that the vast majority of microbes that we know about have yet to be cultured. And I don't mean 80 or 90%, I mean 99.99 something. Right? And every time we go out and look in an environment, we find microbes there that are new to science. This is not uncommon. We get excited when we discover a new species of insect or beetle perhaps, or fish, but the discovery of new microbial species occurs very likely every day all around the world. And the question, it sort of begs the questions, well, what are all these microbes doing? And how are they making a living? And what's their role in our biosphere? So that's the focus of our research. So try to figure out ways to understand what uncultivated microbes are doing in the world around us. So what I'd like to do, if this is all right with you, is give you a presentation here on some of the approaches we've taken, some of the systems that we've studied. Try to keep it at around 40 or 45 minutes so that we can have a nice time talking together at the end and having a nice sort of question and answer session. So if that sounds good to you all, I'll go ahead and get started. Anybody want to take a guess at what this is on the right-hand side? Not Tubeworms? I heard someone say vent. It's actually chalk. It's about a 60-foot tall chalk tower in the mid-Atlantic. You may have read about this. It was called the Lost City. And it's a structure literally made largely of chalk that is made entirely by microbes. More accurately, I should say that the skeleton is formed by geochemical processes, and microbes are contributing to the reshaping and reforming of this. So it just goes to show that despite being minuscule in size, the impact that they have on our biosphere and on our geosphere even is really quite tremendous. Well, I want to start by diving into some geology real quickly. During the late 1960s, there was still a vigorous debate about plate tectonics, right? And whether or not we could find signs of plate tectonics in the deep ocean. Now, you may all be wondering, well, what does it really have to do with microbes? Well, actually, it has everything to do with microbes, because it was the desire of geologists to study these processes on the seafloor that led to the discovery of hydrothermal vents, and really fundamentally changed the way we think about life on Earth. What geologists wanted to do was to go to the seafloor and look for signs of plate movement, something that we, I think, take for granted today. And it was their intention to go to one of these putative plate boundaries and look for signs, again, of plate spreading, which would include volcanoes. We see volcanoes on land, so they said, well, why don't we see volcanoes underwater? Hot springs, things of the sort. And there were a number of candidate plates, this little Cocos plate here and the Juan de Fuca plate there, that were small and easy to get to. And at the time, the United States Navy had recently invested in the Alvin submersible, which many of you have probably heard of. And so it was by the sort of insistence of geologists and the availability of a small deep diving submarine that we were able to go down and explore these spreading centers. Now, the availability of Alvin really fundamentally changed the way that we do science in the oceans. Until we had access to deep diving submarines, a lot of what people tried were so I'll leave it at that. So a little quick introduction to Alvin. Who here has heard of Alvin? All right, so we'll keep this brief. But as you know, it takes 3 people to the seafloor. You might not know that it's been featured in over 2,000 scientific publications and is arguably the most famous submersible of all time. It's something that I've had the privilege of working a lot with, and I have to say it's an exhilarating ride to be dropped off the back of a ship and to head to the seafloor to do your science. When these geologists first took Alvin to the seafloor in search of these underwater hot springs, what they found were these strange structures that looked like chimneys, and they nicknamed them chimneys, or called them more specifically black smokers. And you see this kind of plume coming up here, which, as many of you probably know, is dissolved metals coming out of solution as the hot water cools and gets oxidized. Well, here's a little video just to give you a sense of what it looks like from the portal of a submarine. It's really, really impressive. And one of the first things that they really wanted to understand is, well, what is this? How does this take place? What really drives this geological, venting? And what we understand now is that these vents behave a lot like coffee percolators. Now, I'm bringing this up for a reason. Be patient with me. I promise you we'll get back to microbes. But it seems like a number of you here may be familiar with coffee percolators. When I talk to my students now, it's sort of a lost metaphor. But really, you have this heat source, a magma chamber or something that's heating seawater and making it less dense and buoyant, and it comes up through the path of least resistance and emerges from the seafloor. That process actually entrains cold seawater in areas around these vents, areas that we call recharge zones, that actually bring cold, nutrient-rich water into the sediment. And we're going to come back to this shortly. But while the geologists were incredibly excited by what they found there, it was really the biologists who were overwhelmed and just floored, not only by the novel life forms they discovered, but also the abundance of life. That's really what I want to focus on, is that these hydrothermal vents are not only interesting from a geological perspective, that is to say they informed us about plate tectonics, gave us a better sense of spreading rates and so on. So geologists were thrilled, but the biologists were just beside themselves. And nobody expected to find this much biomass on the seafloor. Now, how many of you are familiar with the deep sea? Do we find a lot of life there in general? No, and the reason is there's no light. There's not a lot of what we call primary productivity taking place in the deep ocean. So the conventional wisdom was that you have a bunch of dead stuff falling from the top, you got a few kinds of animals, and that's it. We went to these hydrothermal vents, and we found enormous communities of organisms. And this is a picture, it's not too old, taken a little while back of a clump of these giant tubeworms. And you can see that it's surrounded by clams and crabs. I mean, there's an awful lot of biomass around these hydrothermal vents. These are some of the giant clams that we find down there. And it's not just tubeworms and clams. We found new species of blind crabs. We found limpets. We found worms here that have very strange bristle brushes on their back, which we now know is an area where these worms culture microbes. We found these little scale worms. I mean, there's an incredible diversity of organisms, from kind of creepy looking fish to blind little squat lobsters. We found that life thrived around these underwater hot springs. And it also became apparent that these communities were dominated by just a few species of really abnormally large animals, right? Giant tubeworms and giant clams. These tubeworms their tubes can reach 6 foot in length. And they're really quite large. They're about the diameter of a $50. Here's a clam. You can see someone holding, these are massive, massive animals. And all of these, by far and away, all of these abnormally large animals that lived at hydrothermal vents had one tiny little thing in common, and that is they are symbiotic with bacteria. And more specifically, they're symbiotic with bacteria that we call chemoautotrophic bacteria. Have any of you heard this term before? Yeah. Okay. So essentially, these bacteria do what plants do, only they use chemical energy instead of sunlight. And they form these associations with these animals at hydrothermal vents. And they actually form the entire base of the food web at hydrothermal vents, which is really amazing to think that you have entire ecosystems that flourish. And I don't just mean barely eke out a living, but I mean really flourish. And they thrive largely independent of the sun. Now, in all fairness, these animals that I've showed you all depend on oxygen, which is essentially a photosynthetic byproduct. But a lot of the microbes we find down there live devoid of oxygen. They're perfectly happy without it. And to see that they could support communities of this size is really fundamentally changed the way we think about life on Earth. Now, many of you probably were here for Colleen Kavanaugh's talk, in which she probably spoke quite a bit about these tubeworms. Colleen was one of the scientists who discovered that they actually have these symbionts. And as a young graduate student, I was absolutely thrilled and impressed by this and thinking, wow, here's this worm that makes a living by associating with bacteria. But I really wanted to better understand what does that really mean? How fast do these worms grow? What does that tell us about their life history and their ecology? I wanted to start by asking one of the most basic questions: how much productivity do we see resulting from these symbiotic associations? And I remind you that these worms have no mouth or no gut, right? They're essentially sealed off bags that culture bacteria inside them. And they provide the symbionts with all of their nutritive requirements. The symbionts in turn fix inorganic carbon to sugars and feed themselves and their host. And so it was this question of net primary productivity that nagged at me, because we go down there in these submarines and we see these enormous aggregations, but we really had very little idea as to how long it took these to form. Are we looking at tubeworms that have been growing for 100 years? Or is this 6 months of growth? And trying to crack that, trying to address that question was a formidable challenge. And what it set us to task to do is to develop ways to keep these animals alive on board ship, in high pressure vessels, and to measure the amount of material that they took up and released so that we could really constrain the activity, the primary productivity of these associations. And this is just a kind of a quick little cartoon to give you a sense of how that works. We recreate hydrothermal vent water. We use high pressure pumps to pump into these high pressure aquaria. And then we send the effluent stream to a mass spectrometer for analysis. So for any of you who've worked with mass specs, you know that they operate at high vacuum. This was a bit of a challenge to figure out how do you take water at 4,000 pounds per square inch, depressurize it, and introduce it into the high vacuum of a mass spectrometer. So it's kind of a little photo of one of our smaller setups, and I insisted on having windows in our little pressure vessels so we could see the little tubeworms living inside there. Interestingly, we also found that we could keep them alive for up to 3 months in the laboratory. Why we can't get past 3 months is still beyond us, but some of the other species we've been able to keep alive for years now. The catch is we have to keep them under pressure at all times. Yeah?
Why do you have to keep them under pressure?
That is a great question. Anybody want to take a stab?
Since with your environment,
Biochemically, anybody have a sense of why that is? Largely it has to do with membranes. All of our membranes have to maintain a certain homeoviscosity, right? You want them to be a little fluid and a little rigid, and these are adapted to exist at those pressures and temperatures. And pressure acts to confer stability. So if we try to keep them at 1 atmosphere instead of 300 atmospheres, right, or 16 psi instead of 4,000 psi, their membranes don't function properly and they die. So that's why we have to go to great lengths to set up these elaborate systems.
Are they fluid?
Yeah, absolutely. That's exactly what it is. And you can counteract that by reducing the temperature, but we're already close to freezing, so there's very little room there. So what was really absolutely awe-inspiring is that when we looked at the amount of carbon fixed by single tubeworms and we extrapolated it to a clump of tubeworms, we found that these aggregations of tubeworms, of Riftia, are some of the most productive ecosystems on the planet. And I don't just mean kind of productive, I mean really productive. They're more productive than kelp forests. And we always think of kelp forests as being these really highly productive ecosystems or seagrass beds. And when you compare Riftia to other chemoautotrophic systems, like bacterial mats, you find that they're just almost an order of magnitude higher in productivity than some of these other systems. And they're almost as productive as these coastal upwelling zones, which are some of the most productive habitats on the planet. Why is that important? Well, because we're talking about an ecosystem that is largely uncoupled from the sun. We're talking about primary productivity that at its most kind of basic level can occur without dependence on photosynthesis. You're talking about an entire ecosystem where chemical energy in the form of sulfide is being turned into, essentially into biomass. So this is really, really exciting, and again, fundamentally changed the way we think about life in the deep ocean, and this is all thanks to the enormous physiological capacity of microbes. This is sort of an amazing shot that, whoa, where'd it go? There it is. That still blows me away to this day. This is an area about 1 meter square on the Juan de Fuca Ridge system. This is the side of a hydrothermal vent chimney, and it is so packed with biomass that you can't even see the bare rock underneath. And all of this has been supported by chemoautotrophic bacterial growth. So really, it's important. I'm trying to drive home here is the idea that this chemoautotrophy, this capacity to use chemical energy to fix carbon, can support entire ecosystems, and that these bacteria do in fact form these associations with worms and clams and mussels, and it's important to note that they don't kill them. And in fact, they feed these worms and they form these nice symbiotic associations. But we have to be careful about this mindset of thinking that this is some altruistic event, right? Or it's an altruistic association. In fact, microbial symbionts have a much easier life inside of a host than they do outside of a host. Because one of our hypotheses, right? One of the things that I really tried to wrap my head around is why is it that these tubeworms are so much more productive than their free-living counterparts? Well, the answer, I believe, our idea is that these tubeworms act as nutrient reservoirs. See, microbes are single cells, and they can store a certain amount of material, substrates and whatnot, inside their cytoplasm or inside organelles, but they're really quite limited, aren't they? It's a single cell, and they're subject to currents or grazing or any number of other things. And for a chemoautotrophic microbe that needs both sulfide and oxygen, which don't coexist in the environment, right? They spontaneously react. Life outside of an organism involves trying to, get some sulfide from some of the hydrothermal vent flow and get some oxygen from the cold bottom seawater. And so there are even microbes that form these long, skinny sheaths inside which they move between sulfidic zones and oxic zones, and you can imagine how much energy they expend. Well, these animals that host these symbionts typically have vast pools of hemoglobin that bind not only oxygen but sulfide. And so these animals act as nutrient reservoirs, enabling these microbes to adapt to an environment where they're not nutrient limited, and they can really, really crank through substrate and be highly productive, right? So it is arguably beneficial for a microbe to form an association as well. And there are some similarities to pathogens, right? And in some sense, the line between pathogenicity and symbiosis can be fine, and it seems to be getting finer and finer, especially via some of the mechanisms by which organisms acquire their symbionts. And we can talk a little bit more about this later if you'd like.
Do you have an estimate, like what % of the Earth's biomass is in these things?
Well, that's a wonderful question. What % of the Earth's biomass is actually tied up in chemoautotrophic communities? We don't really know. And in some sense, I can guess. I can tell you that per square meter that these things are fixing a tremendous amount of carbon, and we can guess as to how many vents we'd find along the ridges. But we've only explored a very small fraction of these ridge systems. And in addition, we're beginning to find that entire regions of our world's oceans are hypoxic, if not entirely anoxic, right? They're low in oxygen and may have enough sulfide there to support free-living chemoautotrophic activity. And while the rate per individual microbe may be lower, as I showed you in that plot, if you think of the free-living microbial communities, that is a tremendous volume of ocean. So I think we really don't know, but I'm willing to bet you that it's far greater than we've attributed it so far. I think it'd probably be less than 10%. And so, that's the interesting thing about thinking about this in the context of photosynthesis. So much of our productivity is photosynthetically driven. But the fact that you can sustain communities even approaching a few % of total productivity really tells us a lot about the possibility that chemoautotrophic systems could exist or existed here on Earth long before there was oxygen, or might exist on other planets. So, Well, another thing that I became interested in, which is a kind of a nice follow-on to this question, is, what can we learn about the microbes that live in the environment? I mean, I have an interest in symbiosis, but I keep finding myself, thinking about these hydrothermal vent environments and what they represent, and how they might be a good place to study microbes living on the very edge of biological existence, if you will. Hydrothermal vents are some of the most host some of the sharpest geochemical and temperature gradients on the planet. Here's a picture of just a portion of a vent. The smoke and mirrors tower is 70 meters tall. All right, so this is about 210 foot tall structure. I can only fit 10 meters on the slide, and kind of give you a sense of what it looks like. It's this craggly, knobby vent that has extremely hot water flowing inside it. It's on the order of 300 or 350 degrees Celsius, right? And the walls of this chimney are just a few centimeters thick, maybe 10 or 20 centimeters, right? That's not a lot. And on the outside, you have seawater that's at near freezing temperatures. And we know that a lot of microbes colonize the outer surfaces of these, but a colleague of mine, Deb Kelly at the University of Washington, she studies the geochemistry of these vents, and she and I became very interested in this question of, how many microbes live inside these chimney walls, and what's the kind of hottest temperature that they can exist at. Yes?
How old the vent like that?
How old is this structure?
Yeah.
I don't know. I would guess it's probably on the order of maybe 10 years old, maybe 20. They grow really fast. In fact, on this next slide, this is a structure called Fin, and it was in the same field as the smoke and mirrors vent that I just showed you. But Deb Kelly and John Delaney at University of Washington actually worked with the American Museum of Natural History to build an underwater chainsaw, I kid you not, and go down, and they cut this off, wrapped it in a cage, and brought it to the surface. And if you're interested in seeing it, it's at the American Museum of Natural History in New York. So you can actually go see this enormous section of a hydrothermal vent. Well, from the material that they recovered, a colleague of mine at the University of Massachusetts Amherst isolated and cultured, I want to emphasize the word cultured, the most thermotolerant organism we have in culture to date. In fact, it grows at 121 degrees Celsius. Anybody recognize that number? Autoclave. So he kept trying and trying and trying and trying to autoclave his material and sterilize his samples, and he kept having some contaminants. And for the life of him, couldn't figure out what was going on until he thought, well, wait a minute. What if what I'm doing is selecting for a single bug? And as it turns out, that's the case. It's an iron-reducing organism. He published on it in 2003, and this bug again grows, has a nice growth optimum, a generation time, at about 121. Now, it doesn't really do very well at temperatures above 130, right? But this is impressive that we've been able to culture this organism that grows at 121. However, I refuse to believe that the most thermotolerant organism on Earth happens to coincide with our autoclave temperatures. So, kind of begs the question, what is the upper temperature limit of life on Earth? And in studying microbes, we've begun to realize that some of the most extreme microorganisms are not actually bacteria, but are these archaea. You've all heard of archaea, I presume? And there are these core archaeotes that are of recent interest to us scientists because they keep turning up in some hyperthermophilic environments. So we really wanted to look not only for bacteria, but also for archaea. And as I mentioned, there are really, really sharp gradients here, not only in temperature, but also in chemistry. And what we wanted to do is study the growth of microbes along this gradient. And so Deb Kelly said, well, you know what? I think there's a way we can do this. And over lunch, we sat down, we sketched out a design for this hydrothermal vent incubator. We had to make this, the principle here was to go to a hydrothermal vent chimney, to drill into the side of it, and to insert an incubator here that would allow microbes to grow inside the incubator. But we wanted the incubator to be exposed to different temperature and chemical milieus. So we had to have an incubator that spanned the length all the way from the hottest water to the coldest water, that had baffles inside of it so that we could separate fluid flow. We didn't want hot water from here contaminating our cold water site. We had to pack it with sterile substrates, and we had to make sure that it didn't dissolve. Well, the first one we deployed dissolved, and it was a test. Incubator that I made out of aluminum. We knew it was going to be bad, but what we didn't know is it was going to be that bad. We put it down for 24 hours and went to recover the incubator and pulled out just a little stub. And so we realized, okay, so no more fooling around. Let's do this right. So we manufactured or fabricated these titanium incubators. There are temperature probes inside here. That explains this big box. It collects all the temperature data and stores it. And it allowed us to look at the growth of microbes. Here's a picture of it underwater. I mentioned Finn that had been chainsawed off. Well, this is its sister vent, Rowan, that has also been chainsawed. You can see this is new growth after about 2 years. This one is especially slow. For some reason, Finn seems to be growing faster, so we don't really quite understand that. There's our incubator, and there's the wires coming out here to what we call the pig. This is just a titanium bottle that allows us to put all our electronics inside there and keep them at 1 atmosphere. I have a little video to show you of how we did this. This is a remotely operated submarine called Tiburon. It's at the Monterey Bay Aquarium Research Institute. And what we did is Mbari built this custom drill rig. And this fits on the front of the sub. You'll see the drill barrels rotating, and they're going to start drilling. Now, this isn't like your Black & Decker drill at home. It's really, really, really slow. And depending on what we're trying to drill through, this can take a long time. This one took us probably about an hour and a half. Some of our drilling efforts took about 8 hours. So you sit down there and you wait. But what's really exciting is not only did it drill a hole, but it also recovered a core. And so we have what I would call a frame of reference, right? So we can look at the microbes within those minerals to have an understanding of where they're from. Now we have a temperature probe on board the submarine. We're taking the temperature here. The temperature on the innermost part of this drill hole is 250 degrees Celsius. And then on the outermost part, again, is near freezing seawater. The ROV pilots use their manipulator to set the pig up on top of the structure. And you can see things go really, really slowly. So they're going to grab the incubator gently. And believe it or not, they're actually quite good at this. So they get the incubator deployed, and one of the things that I'll point out in a second here is that it was designed so that it had these baffles. They're going to squeeze those pins together, and that's actually going to open up the holes and allow fluid flow. So we can actually pack them, keep them mostly sterile, mostly clean, and stick them in the incubator. When we came back a few days later, you can actually, this is footage from a few days later. You see this white streak there. That's precipitation that had newly formed. And actually, within about 2 weeks, this incubator was sealed into the side of this chimney. So it just, which is wonderful news. It just enclosed it right in. So this experiment is really designed to help us better understand where microbes grow, what's the upper temperature limit, to tell us something about the chemical conditions at which they grow, and to better inform us as to how organisms make a living in these hydrothermal vent chimneys. Now, our initial results, which I'm showing you here, is some mineral wafers that a colleague of mine at the University of Washington prepped and polished up, and then using a fluorescent microscope, looked at biofilms on these mineral wafers, found signs of biofilms growing at temperatures around 140 degrees Celsius. I started looking at DNA that I recovered from minerals formed inside this chamber, and was recovering DNA from temperatures greater than 150 degrees Celsius. So we think that somewhere around there is the upper temperature limit of life. What I haven't shown you here, though, is what I think is the most interesting observation. Our temperature data suggested that, in fact, there was a tidal signature in, we observed a tidal signature in the temperature data. which tells me that as a function of tidal forcing, you have sections of the incubator that are either hotter or colder. Now, here we went into this thinking that we would find some temperature, 155, whatever it is, at which we would find microbes, and then some temperature, call it 160, at which we wouldn't. But, not surprisingly, nature is more complicated than that. And we know that there are a lot of organisms that are quite capable of forming spores and surviving exposure to high temperatures. And so what we began to wonder in looking at this tidal signature is if we see a bug at 180 degrees, did it actually grow at that temperature? Because what we have to figure out is at that point in space where we isolated that microbe, what was the coolest temperature? Well, if it's 120 degrees, well, maybe it's growing at 120, forming spores, and enduring 180, and then when it's cool again, undergoing cell division. Yeah?
So what was the amplitude of the tidal signature? So like 60 degrees C?
The amplitude was, in some cases, I believe as high as 40 degrees C. So that's pretty impressive. So now we have to stop and think about how we're going to address that. So what do you call the upper temperature limit of life? Right? Is it the temperature at which spores can survive? Or is it the temperature at which you see cell division? So this already challenging project became even more challenging. And what we're doing now is really trying to push hard on refining our analytical techniques to better constrain what we believe is the upper temperature limit of life on Earth. So I wanted to kind of close with that and then have a discussion with you all about another exciting recent finding. But I just wanted to emphasize that it is so easy for us, all of us, even microbiologists who work around microbes day in, day out, to have that bias to think that we often associate microbes with disease, or we associate microbes with somehow being nefarious. Right? We're always thinking, well, there's got to be some virus behind this or some microbe. Well, what it really comes down to is microbes are trying to make a living like the rest of us, and it's important to remember that most microbes on Earth are actually keeping our planet alive. They're responsible for the biogeochemical cycles that keep our air clean and our water clean. And again, microbes can support entire ecosystems without dependence on the sun. This is really, I would argue, one of the greatest findings of 20th century science is that investigators such as Colleen and others who went out in those early days really recognized that they were looking at novel and exciting ecosystems that were fundamentally different than anything we'd considered in our thinking about life on Earth. And I think that what we have to start doing in our kind of current and future directions in studying microbes is begin to try to better understand what the role of these vast numbers of uncultivated microbes, what is their role in governing the health of our planet, and how do they dictate not only their own ecology, but our ecology and those of the animals that live in the biosphere. And with that, I would like to thank you for your time and see if we can have a nice chit chat. So thanks again. Any questions on this? Yeah.
My understanding of these vents is that they come and go. So they stop putting forth hot water, they collapse, whatever. Mm-hmm. So how do the microbes, which are so important to the ecosystem, show up in the next new vent place? I mean, any ideas about, do they just, how do they populate it with a place that you don't even know is going to exist yet?
Right.
What will happen?
That's a great question, and one that we often struggle with. The question is, how do microbes go from one vent to another? These vents are ephemeral, and it is, it's not as though microbes can necessarily swim long distances. They're subject to the whims of currents and everything else. So how does that work? Well, there's a saying among microbiologists that everything is everywhere, and the milieu selects. And this notion was put forward actually quite a while ago now, and it is something that we as a community are really trying to better understand. The work of many microbiologists, both here and and at other universities, including Colleen, have really tackled this question of biogeography. Right? That is, do you actually see patterns within microbial genomes that tell us that there are distinct populations in certain regions? So her interest, for example, is looking at the symbionts found in mussels, and to ask the question, do these symbionts, are they related to the symbionts that you find a kilometer away? What about 100 kilometers away? And what we're finding is that there are definite biogeographical provinces, but the important thing to note is that the story with microbes is rarely clear. So we'll find that if you look at one set of genes, it tells you that these populations haven't been mixing. But then if you look at a different set of genes, you actually get a very different story. And I think this is because microbes are quite good at exchanging genetic information. And so there really isn't an easy marker, right? I mean, microbes are swapping genes left and right, not only among themselves, but also through viruses and other sort of different factors. So it's a tough nut to crack. Yeah?
What can you tell us about chemosynthetic microbes that don't live in extreme conditions?
There are a lot of chemosynthetic microbes that don't live in extreme conditions, including in a lot of your water heaters. The requirements for, well, let me start again. Typically, when you go to an environment where you find modest or more modest amounts of organic carbon, for example, you begin to see signs of what we're going to call lithotrophic growth. That is, microbes that make a living off of converting the inorganic material around them into organics. Conversely, when you go to very organic-rich environments, you see the production of hydrogen sulfide, and you begin to see microbes that use sulfide as an energy source, and they'll also fix inorganic carbon. But most of the work that's been done on chemoautotrophs have actually been done in fairly, I would say, kind of fairly modest environments. A lot of hot springs where they're not near boiling, but they're 30 or 40 degrees Celsius. And that's a lot of work has been done because those environments are a lot more accessible. But they are ubiquitous, and anytime you catch a whiff of that rotten egg smell when you're walking by a sewer or rummaging through your compost pile, chances are there's some chemoautotrophic bug that's making a living off of that. Yeah?
Has anybody studied the phylogeny of those tubeworms, who they're related to? How did
Yeah.
they get there?
Yeah, the phylogeny of the tubeworms is really interesting because when they were first discovered, they were described by a fellow named Meredith Jones, who actually lived and worked around here, spent a lot of time down on the Cape, and he said that that these probably represent, they might even represent their entire, there might be a separate phylum altogether because they're so unique. In more recent years, I think studies have pretty clearly demonstrated that they're in fact related to annelids. And maybe I'll flip back here and show you a picture. That one. Believe it or not, if you look at them genetically, they actually appear to be related to annelids, and they're their own, I think they are their own family. And this gill structure of theirs was likely derived from a segment, as was this section, as was this section. And where you find evidence of segmentation is in this tiny, tiny little kind of tail here called the epistosome. So if you look in there, you can actually see morphological signs that this, in fact, was at one point highly segmented. So.
Do they know which annelids they're most closely related? I mean, I'm trying to get where did they come from and how are they related to each other at the different vents?
Well, they're all, again, within this family, Syboglinidae. And all of these tubeworms that you see, whether it's Riftia or the hydrothermal or the hydrocarbon seep tubeworms, which we call Lamellobrachia, and even these Poganophorans, which are the shipworms. Well, they are small worms that live on ships or other deadfalls. They're all closely related to one another, and they seem to fall within the annelids again. But in terms of their nearest, I guess typical relative, not really sure. But like many organisms we have discovered in the deep sea, the discussion about where they fit is ongoing, and that's pretty much the deal. Yes.
You got, yeah.
Yes.
You said that you had, you showed us the HPRS and where you had, you measured productivity. I was just wondering, when you had these tubeworms and the symbionts up here on ground level in the laboratory, was any other kind of biochemical work done? Do you know anything about what's the biochemistry in there? How is it happening at such high pressures? And is there a difference in the protein structure? What are we looking at?
Yeah, that's a great question. The question was, well, what do we know about the biochemistry of these tubeworms? Most of the initial work was done on the hemoglobin back in around 1980. So they discovered these vents in 1977. The first serious biological expeditions occurred in around 1979. The descriptions of the symbiosis occurred in 80 and 81. And at that time, grad students and professors were really just looking at these worms and saying, well, let's start describing the most basic features. And one of the things that was obvious is they had a lot of really bright red blood, and which gives them this red color. And the idea is, well, these things live in hydrothermal vents. There's a lot of sulfide there. The worm, of course, is an animal, so it needs oxygen. Maybe this is just, a lot of blood to bind a lot of oxygen, because maybe the oxygen concentration is low. Maybe there's not a lot of oxygen. Well, it turns out that all of those things are true. They do have a lot of hemoglobin. It has a very high affinity for oxygen, but it also separately binds hydrogen sulfide. Now, as I mentioned before, hydrogen sulfide and oxygen are highly reactive and don't coexist. They're not stable. You could take sulfide and oxygen in a little beaker and shake them up, and in a few minutes, sulfide's largely oxidized. Well, this is one of the amazing adaptations of this worm, is that it can take up oxygen and sulfide, bind them, stick them on different parts of the protein, and they don't react. So again, it can transport both oxygen and sulfide to its symbionts. That's one of the biochemical adaptations that have been well studied in here. We have looked at the effect of pressure on enzyme activity. It's in particular proton ATPases. It's pretty clear that that these organisms, have evolved at these pressures, or at least are adapted to these pressures. It doesn't seem that enzyme function is, in many cases, greatly influenced by pressure. And so we've sort of ruled that out as the cause of death when we try to keep them at 1 atmosphere. I really do think it comes down to membrane fluidity. But yeah, there's been a fair amount of work that's been done both here and in France. So, yes, sir, sorry, in the back.
Yeah, I had two questions, but you've answered one of them already, so that's okay.
Sorry.
The second one is the conversation about how we can talk about microbes as having the positive impact of microbes.
Yep.
And I was thinking, here we have a bunch of organisms that are living separate from the ecosystem in which we live. Mm-hmm. We have a huge problem of what to do with all our excess carbon dioxide. Is there a way we could pump it down there and let them take care of it, take it out of the atmospheric ecosystem, and get it lithified, because that's what we want to do?
That is such a good question. I think that there's a lot of interest in sequestering carbon in the deep ocean, not necessarily because of chemoautotrophic microbes, but because at those pressures and temperatures it's quite stable, relatively speaking. One of the things that that a number of us have considered is, well, what would the effect of carbon dioxide be on the activity of chemoautotrophic microbes? I mean, if you're going to put this down in the deep ocean, you're going to sit atop of a sediment that will go anaerobic and will thus begin producing sulfide, and you're likely to encourage the growth of chemoautotrophic microbes. I think that is a good idea, but I think doing that in situ on the seafloor is more challenging than one might think. And the reason is, to give you an example, is even if you could demonstrate in the lab that a chemoautotrophic microbe could fix carbon at some astonishing rate, if the partial pressure of carbon was so high, or the concentration of inorganic carbon was so high, there are other covariables. I'll share two. One, of course, is pH. And if you're forming carbonic acid, the pH is going to drop quite rapidly and dramatically. And the second one is carbon dioxide. This is sort of an experiment we're just setting up in the lab now. Carbon dioxide is the precursor to methanogenesis. Microbes make methane, and they use CO2 to do it and hydrogen. So you put down a lot of carbon dioxide, but what if you start getting a lot of methanogenesis? Well, that's not good, because it's a more potent greenhouse gas, 65 times more potent than CO2. So at the Monterey Bay Aquarium Research Institute, they're actually doing quite a bit of work on this topic. And they're trying to find ways to sequester carbon in the deep oceans that's economically relevant and environmentally somewhat friendly, I guess, right? Environmentally acceptable. And I really personally think that, you know, we as a species, right, humankind has to recognize that there is no quick fix, and that whatever we're going to do is going to have an impact somewhere. We just have to be judicious about, you know, what kind of impact we want to have. So there's a lot of work going on, and I think it's an interesting idea, but I would argue that this would be best done in some kind of reactor. And to kind of close on this note, some estimates, just sort of jog my memory here, some estimates have suggested that in order to sequester enough carbon dioxide in the deep ocean, we'd need a pipe that is the diameter of a 4-lane highway running into the deep ocean from someplace like Hawaii, wherein we would be concentrating CO2 or capturing CO2 from the atmosphere and piping it down as a liquid at a rate of 3 meters a second. So that's a lot? Right. So there are some challenges, but
Well,
yes.
When I had asked before about chemoautotrophic living at ambient temperature,
Yep.
I was kind of going down that road of perhaps farming them in
Yep.
such a ways to sort of recycle this, use some of our waste and CO2 pumped in there, and then get some methane out.
You know, that's a really good idea. And the Department of Energy has, to their credit, spent quite a bit of money over the last 60 or 70 years in trying to develop ways of converting carbon dioxide into methane sort of biologically in some commercially relevant manner. And while we have yet to have some kind of reactor in which that can happen, you know, some sort of, I'm going to say, we have yet to come up with a turnkey system wherein you would put carbon dioxide and get methane of, you know, X quality and Y concentration. I do think it's fair to say that people have made a lot of progress on capturing methane from, say, landfills where this process occurs naturally. And I think that that's a very, very good idea and a very underutilized resource that is actually arguably carbon neutral, right? Because it's all about, if you've got CO2 in the atmosphere making methane and you burn it to make CO2, right, independent of how bad things are now in terms of the amount of CO2, but that is a carbon neutral loop. So if there's a way we can achieve that, then I think that would be quite good. So, yes?
Because of the limitations of being able to see very far, every time I see pictures of these undersea vents in the communities, you know, you're only looking at like one smoker. I have a hard time wrapping my head around how extensive are these communities. Are there very many of them? What are we talking about in terms of how far they spread? Is it, you know, we're looking at one chimney, or is it meters across, or?
Scale. It's hard to have a good sense of scale. Yeah, you're absolutely right. And so the question was, you know, how big are these? How many of them are there? How diverse are they? I mean, well, I guess not how diverse are they, but yeah, you know, you get these snapshots that are neat looking, but you really don't have a sense of how big they are. The first hydrothermal vents that were discovered were, interestingly, perhaps ironically, near the Galapagos Islands. And they were these tubeworm communities. Now, the vents around the Galapagos and at the East Pacific Rise are relatively small. A typical structure, smoker you might see, would be about the size of this podium. And you would see tubeworms around them, probably in an area about the size of this stage. I think it's fair to say that's pretty common. The fields where we do most of our work, there was a field we called Rose Garden, right, where it was just expanses of tubeworms, probably occupied something the size of this room in terms of area. And you probably had structures within it that were maybe 1 meter or 2, maybe 5 meters tall. There are some structures there that are maybe, probably at most, maybe about 7 meters tall. On the mid-Atlantic ridge, we have vent systems that are equal in proportion. The communities are really quite different. There are no tubeworms on the mid-Atlantic ridge. No, people have yet to find one. Lots of shrimp, blind shrimp, that have a weird eye spot on their back, but no tubeworms. The vent field that I absolutely just find most thrilling are those on the Juan de Fuca Ridge system, up off the coast of Washington, where we do most of our work. That's where that smoke and mirrors structure is. But when you go there, it's kind of like flying through some fairy tale castle, because you have immense structure after structure. There's an area called, which what we call the sort of Faulty Towers complex, named after the British TV series, if you're into that sort of thing. And it is just 20, 30 meter tall structures, right? You're talking 40, 60 feet tall chimneys from the base to the top, and they're just lined up in a row. And the Alvin submarine takes a reasonable amount of time to fly from one end to the other. So the fields are several hundred meters in length. So that is really, it's really quite majestic. And when you see pictures like this, right? This represents a community. Why does it keep doing that? This represents a community of animals, again, about 1 meter square. And this was taken about 10 or 15 meters off of the bottom, up on the side of a chimney. So they're really, really massive. And we believe that they're that big because that is a very rapid spreading area. And so they just grow really quickly. But it is hard to get that sense of scale. Yeah.
Do you have any of the tubeworms or shrimps living on the surface in a replicated environment? Has anything been brought up and is still living, alive?
Has anything been brought up and is still living or alive? When I was in California, we were doing that quite frequently because the ship would sail from San Diego and would go down to the East Pacific Rise, which is about a 5-day steam. We do our work, and at the end of the expedition, I'd bring all these animals up and I'd just pack these pressure vessels with worms and mainly worms then. Pressurize them and keep them pressurized. When we get into port, I'd even start recirculating it so I wouldn't introduce nasty, dirty port water. And then while the system was under pressure, I'd seal them off like a big soda can, stick them in the back of the U-Haul in an ice bath, and drive them up to Santa Barbara and get everything set up again. It was a lot of work, but we pulled it off time and again. The tubeworms are a little more difficult to maintain than some other animals as we know now, but they were really exciting because not only could we keep them alive in the lab, but I could watch them grow. So they would grow tube at a rate of about 1 centimeter a day. So if I could afford a time-lapse camera, I would have put one in there and you could have just seen them, kind of go on. But we didn't even need it. You just go in once a day and take a snapshot, and you just, make a little flick book. I mean, they grew that fast. After moving here, it became a little more challenging, and over the last couple of years, we've literally FedExed animals back, but we can't keep them at that high a pressure, and it's a lot more challenging. One thing that is really interesting, though, is that the animals that we recover from the Gulf of Mexico, which are hydrocarbon seeps that are found at about 500 or 600 meters water depth, can be adapted to live at 1 atmosphere. And until just about 2 weeks ago, I had a colony of them growing upstairs in the lab. And then we had to do some experiments, and so it kind of put an end to that. But one of the things I'm hoping to do in the next year or so is work with the Gulf of Maine Research Institute. There's a fellow up there named Ed Seidel, who's a collaborator of mine, and we're going to make a hydrocarbon seep exhibit. So next summer, I'm going out to the Gulf of Mexico. We're going to recover more tubeworms and more of the mussels and animals that live down there, and set those up, and they should live for years. So hopefully, if all goes well, you'll be able to see them. Yeah.
Question about reproductive activity of tubeworms. Are they hermaphroditic? Ian, are you ever worried about, with Alvin, contaminating or carrying up something? Or maybe when you dump Alvin in the mid-Atlantic,
Yeah.
are you going to start to transport something
Yeah, that's-
recolonize?
That's a great question too. So are they hermaphroditic and are we worried about contamination? They are not. They're actually, they have sexual reproduction. There are males and females. They release their gametes through a little tiny pore found right about there. And they sort of free spawn. They're really funny. If I have them in pressure vessels and if I tap on it a certain way, they'll just release gametes. If you shake them, they'll just release gametes. And it's kind of, it's kind of what we call weedy species, do to kind of reproduce, right? They just, anything that may seem like the vent is going to shut down or they're going to be eaten, they're like, all right, let's go. And, but what's interesting about that is it's hard to understand, again, this question of geography. It's hard to understand. I'm not talking symbionts, I'm talking about worms. When their eggs are fertilized in the water column, they form a little trochophore larvae. But we know from looking at worms along the East Pacific Rise that there is genetic mixing all the way from the Gulf of California down through Easter Island. So somehow these little larvae are making their way between, or rather among, the different vents located on that axis. As for contamination, it's a very big concern. Some scientists have suggested that they actually wanted to do transplant experiments. Most scientists said absolutely not, so that hasn't been done. And a number of these hydrothermal vent sites are actually natural preserves. So Canada, for example, has certain vent fields that we're not even allowed to go into. So they're really, we're taking precautions to avoid any issues. Yeah?
On a little bit of a different note, have you ever heard of anyone recognizing any habitat alteration or pollution down there from humans and how these species might deal with, all of our trash or garbage or temperature sensors on the bottom of the ocean?
Yeah, so you're asking, to what degree have they been affected by anthropogenic activities? What about trash or anything like that? I would say that that we scientists actually have the biggest impact. Typically, we don't find a lot of trash from ship trash dumping on hydrothermal vents, probably because it's a fairly small portion of this area of the seafloor. But I can tell you from having been down in Alvin and using robot subs in other areas, I mean the seafloor is actually littered with trash and a surprising amount of plastic. And of course, you're not supposed to dump plastic anymore. And I think most ships go out of their way not to do that. But the thing about plastic is it's probably been down there for decades. The problem, one of the concerns about Alvin, or one of the issues with Alvin, is every time it goes down, it drops weights. And they're iron plates, and it's about 100 pounds of iron plates that are a stack about yay big, and there's 2 of them, and it drops them, and we cruise around and do our work, and then it drops a second set, and we come home. So some of these vent fields, you go there and you see iron plates all over the place. Some scientists, including again my colleague Deb Kelly at University of Washington, has lobbied NSF for some submarine time to go down and clean things up. So there's a modest effort to kind of tidy up our study sites. Yes?
So like the bacteria you looked at in the inner part of the vents, are they bacteria? Are they archaea and methanogens?
A great question. They are in fact methanogenic archaea, is what they look like by and large at the higher temperatures. But then what's also interesting is we're seeing what we think is high representation of heterotrophic bacteria. Now, heterotrophic bacteria do what you and I do, right? They eat organic matter and they either oxidize it or they, some of them ferment it so they don't need oxygen. But to find them in this vent was a little surprising. But check this out. This is kind of neat. A grad student at the University of Washington has been studying dissolved organic carbon in the vents. And it turns out that in some of these vents, there's quite a bit of it. And it's probably because, remember I showed you that coffee percolator? Those recharge zones are areas where there's lots of microbial activity and there's lots of organic carbon. And it's very likely that that's being entrained and passed out through these vents. So seeing heterotrophic activity is not terribly surprising anymore. So maybe one more and then one more.
So I was wondering, you suggested that like spore formers to live through the temperature fluctuations. Are there any spore forming methanogens?
I don't know. It's a good question. I should look into that. That's a really good idea.
Yep. So are there good sources of footage of the biology down there?
Yeah, absolutely. I think that NOAA has a good website. Also, again, my colleagues at University of Washington put together a nice website called Visions 2005. It's from an expedition that we went on in 2005 with, where we did the first high-definition transmissions from hydrothermal vents. So we actually put an HD camera on the robot sub and took HD video on the seafloor, sent it up to the ship, beamed it to a satellite, and sent it to a number of schools and other facilities around the country. The video from that, I think, can be purchased, or I think it's actually free for educators on the website. So that's a good place to look. What do you say, Tara? Well, thank you all very much. I appreciate it.